Okay, so it is a pleasure for me today to be with us, and the title of my presentation is Antiproliferative Effects of Novel Benzothiazole-based Compounds in Pancreatic and Paraganglioma Cancer Cell Lines. My research work in last years was devoted to the identification of novel PPAR alpha antagonists as promising compounds to treat different conditions. You can see in this title of articles that PPAR alpha, a nuclear receptor, is seen as a novel therapeutic target for different tumors, as for example, the chronic lymphocytic leukemia or the kidney cancer, or again, melanoma or ovarian cancer. This is because the PPAR alpha controls many important pathways in the organisms, mainly related to lipid metabolism. And in cancer cells, where the metabolism has different types, often the cancer cells pass from the glycolysis to the fatty acid oxidation. And so to interfere with the fatty acid oxidation could be an important strategy to target the cells, the cancer cells that needs different metabolic pathways. And so the PPAR alpha antagonists in last years are emerging as promising tools to obtain an anti-tumor action because of this metabolic switch in cancer cells from glycolysis to fatty acid oxidation. So the main aim of my work were the following. At first, the identification of novel PPAR alpha antagonists, starting from the structures of fibrates that are classical agonists for this receptor. Then the assessment or the antiproliferative potential of these compounds in cancer cell lines. At the end, we were also interested in expanding the structure-activity relationships of novel compounds. I would illustrate you the strategy that we pursued in our lab to obtain novel PPAR alpha antagonists. And in this specific case, we started from benzothiazole-based agonist. Okay, from benzothiazole-based agonist that we synthesized as heterocyclic derivatives of classical fibrates. In these compounds, we have the carboxylic head, that is a common structural features from the agonists of PPARs. And we decided to change this carboxylic moiety, and we decided to put into novel molecules the sulfonimide portion, because we were inspired by some triazolone synthesized from Eli Lilly, in which the carboxylic moiety was changed with a sulfonimide 1. And the size of substituent in this moiety was responsible for the antagonist behavior of these ligands. Substituents hindered with a good steric hindrance were able to block the receptor in an antagonism with producing an antagonism behavior. So we planned the synthesis of our benzothiazole compounds with this sulfonimide portion, and we changed parts of the molecules, the substituent on the phenyl ring, the substituent on the carbon adjacent to the carbonyl, and the substituent R3 on the sulfonimide portion. Here are reported some of the derivatives we synthesized with this general formula. As you can see, we induced main modification on R1, and we introduced phenyl rings or normal propylic chain, and R2, and we inserted in these molecules different substitution on paraposition of phenyl ring, different substituents with different electronic properties. And we submitted our compounds at a transactivation assay by the PPAR alpha Gal4 methodology on HEC293 cells. At first, we evaluated our compounds of a single high dose at 150 micromolar. Then we obtained some concentration response curves from 1 to 150 micromolars, and we tested our compounds in the presence of the PPAR alpha agonist, the commercial compound GW7647, used at 2 micromolar. You can see in this table, we obtained good IC50 values for our compounds that acted as antagonists for this receptor. In particular, we obtained sub-micromolar IC50s for different compounds, and we decided to submit these compounds to cell viability evaluation. We selected these 5 compounds. As you can see that we have 2 compounds with the phenyl ring in alpha position and 3 compounds bearing the normal propylic chain on the carbon alpha. And these 5 compounds were submitted to MTT evaluation, and we selected some cancer cell lines overexpressing this receptor. In particular, we selected 2 pancreatic cancer cell lines, the lines CAPAN2 and SPC1, and 2 colorectal cancer cell lines, HT29 and SW480. All these lines were previously analyzed into their expression of this receptor. Here you can see the results of this evaluation when we tested at first our compounds at 75 macromolar, and we obtained these results for pancreatic and colorectal cancer cell lines. You can see we used as a reference compound 2 agonists for PPAR, because we want to demonstrate that the agonist on this nuclear receptor didn't affect the cell viability, but only antagonist on alpha isoforms could be efficacious in reduced cell viability. In these pictures, you can see that our compounds induced a decrease of cell viability, and strongest effect was seen for compound 1E, both in pancreatic cancer and in colorectal cancer. At this point, we decided to explore further the activity of this compound, and we tested it at concentration, at different concentrations, ranging from 25 to 150 micromolar for 72 hours. And we obtained these graphs. you can see clearly that the effect on pancreatic cancer cell lines and colorectal cancer cell lines were detected only at high concentration. In fact, you could see that the IC50 calculated were up to 100 micromolar for pancreatic cancer, and at about 70 micromolar for the two colorectal cancer cell lines that were more sensitive to the action of this compound. Despite these values quite high, 1E was interesting for us because when tested in healthy cells, in non-cancerous cells, it showed an activity, it showed a profile of lower toxicity with respect to the PPAR alpha antagonist GW6471. So it was more interesting from a safety point of view. Then we move from our benzothiazole-based agonist, starting from the carboxylic compounds, we decided to obtain novel compounds bearing the benzothiazole portion, but introducing some amide functional groups. In particular, we analyzed the substitution on R1 with alkyl or phenyl groups, and R2, they are all secondary amides, and R2 was an alkyl, a phenyl, or aryl alkyl moiety. We were inspired this time from the structure of GW6471 because it has this amide portion, and it has been demonstrated that these structural features is responsible for the antagonist behavior of this compound, because this part of the molecules block the folding of helix 12 that is responsible for the activation of these nuclear receptors. So we planned and we realized the synthesis of these novel compounds, these secondary amides 2A2G. The synthesis was very simple because we condensed the starting carboxylic acids with the proper amines in the presence of DCC, HOBT, and methylmorpholin in the methylformamide for 24 hours. And at first, we submitted these compounds to the transactivation assay by PPAR-GAL4 technology. And surprisingly, we obtained a moderate PPAR alpha antagonism, also if the action of these compounds was very selective for the subtype alpha with respect to gamma and delta isotypes. And but this antagonism was moderate with the inhibition range of 22 to 75%. But despite this aspect, we decided to submit our compound to cell viability assay by MTT. And we selected to this aim the pancreatic cancer cell lines, some colorectal cancer cell lines, and in addition, we used also two models of paraganglioma. These two models, these two cell lines, PTJ64I and PTJ86I, were developed at my university. They are not commercially available, but they were these lines were immortalized by the pathology group at my university, University of Chieti. So we tested the activity on cell viability, the effect on cell viability, at first at 75 micromolar 2 hours. And here you can see the results on the pancreatic cancer and colorectal. This time we compare our compounds to the effects of GW6471, the commercially available PPAR alpha antagonist. You can see that this series of compounds showed interesting results. All of them were induced antiproliferative effects, and especially most cytotoxic compounds were compound 2B in both pancreatic and colorectal cancer cells, and 2F for pancreatic cancer and 2G for colorectal cancer. But if we compare to the action of GW, you can see that GW was the most cytotoxic compound with inhibition higher than 90%. When we tested our compounds in paraganglioma models, we obtained really interesting results, as you can see from this graph, because all the compounds at 75 micromolar were induced a strong effect on cell viability, a strong reduction of cell viability. In particular, really interesting was the reduction induced by compound 2B that I report here in the figure. It is the compound that shows a phenyl ring in alpha to the carbonyl and another phenyl rings unsubstituted on the amide portion. Okay, we then made some concentration response experiments in these two paraganglioma lines. We use concentration from 3 to 24 micromolar for our compound 2B, and we obtained these graphs. You can see that the IC50 calculated were good. We obtained 8 for 49 for in the first line of paraganglioma and 16.7 for the second line. Interestingly, these IC50 were very similar to that of GW6471 when we tested it on the same lines and at the same concentration. In addition, we submitted this compound also to some clonogenic experiments in these two paraganglioma cell lines, and you can see that we obtained good results in both lines, where our compound 2B was able to affect the clonogenic activity, to reduce the capacity to form novel clones at concentration of 24 micromolar. At this point, we have to clarify the mechanism of action of this compound, because the strong cytotoxic effect shown by 2B doesn't correlate with its PPAR alpha antagonism potency. So the question we should ask is if this cytotoxic effect is only mediated by PPAR alpha, or if other biological targets could contribute to this effect. We tried also to expand the structure-activity relationships of this this novel compound of 2B. We take 2B as our lead compound, and we perform some modification in two parts of the molecules, as you can see here. We studied the influence of substitution on the carbon alpha, of the alpha position, and then we pass to insert some substituents on this aromatic portion to study the influence of this part of the molecule on antiproliferative activity. Briefly, here you can see the results, the preliminary results obtained when we changed the substitution on the carbon alpha. We inserted a normal butylic chain. In these normal butyl derivatives, you can see we took the aromatic ring on the secondary amide, or we spaced it by different linker with methylene units. Briefly, we submitted these compounds to study on 3 pancreatic cancer cell lines and 2 paraganglioma models, but the results were not so interesting because, as you can see from this table, the reduction of cell viability was quite low, with the exception of compound 1 on paraganglioma lines and compound 3 on 2 of these pancreatic lines. But we reputed these compounds not so excellent in their profile, so we passed to study the influence of substitution on the aromatic portion. We retained in this case the aromatic in the carbon alpha, and we make substitutions in this aromatic ring. And as you can see in this table, we synthesized some para-substituted derivatives by introducing substituents of different electronic behavior. The same substituents were placed in meta in compound 4G2L. And then we tried also some desubstitution with these desubstituted derivatives for M2P, where you can see we introduced essentially some halogens in different positions. The synthesis of these novel compounds were really simple. We obtained compound 4, starting from ethyl mandalate, racemic. Then we we reacted the ethyl mandalate with methyl chloride into ethylamine. The ester 1 was submitted to nucleophilic substitution with 5-chloro 2-mercaptobenzothiazole. Then we hydrolyzed the ester in basic condition by sodium hydroxide, and the acid 3 obtained was directly coupled to substituted anilines in the presence of the CC-HOBT and methylmorpholine in the methylformamide for 24 hours. Here are the results of this novel series of derivatives. At first, we submitted these compounds on MTT on 3 pancreatic cell lines, SPC1, BXPC3, and CAPAN2 at the high concentration of 75 micromolar for 72 hours. And you can see that we compared our results to be taken as lead compound. And you can see that in all the 3 pancreatic lines, we obtained improved activity activity and improve antiproliferative activity for several of our compounds bearing electron withdrawing substituents both in meta and para position. At the contrary, when you put in our when you put the methoxy substituent in meta or in para, we observed a decrease of activity, as you can see in compounds 4A and 4G. About the substitution, the substituted derivatives were not so, as you can see, particular for compounds for N, O, and P that showed a decrease of activity with respect to B. In the two paraganglioma models, we observed a very similar behavior, always with compounds with electron withdrawing substituents being more efficacious, more active, and the methoxy substitution and the desubstitution were detrimental to activity. From this screening, we selected 9 compounds of this series, and we calculated the IC50 in these cell lines, so the 3 pancreatic models and the 2 paraganglioma lines. And you can see that we obtained quite interesting results with compound 4D that showed the best cytotoxic effects in all lines, and compound 4L that was particularly interesting for us because it was not the best regarding the cytotoxic effects, but it was the most safe when we tested it on non-cancerous cells. I don't report here this data on non-cancerous cells, but 4L was the most safe compound with respect to the others that showed a toxicity also on healthy cells. Only a brief say about some studies we are performing at this moment of target prediction, because we think that the antiproliferative effects that we have seen in our models are not correlate to the activity on PPAR alpha. So we are in the search for novel targets that are the targets for our compounds. So in this study, in silico study, we are considering different tools, as you can see here, with target prediction, muscles search, different programs freely accessible on the web. And we are filtering results by considering only human binding protein. So now we are analyzing the data, and we hope that we identify some interesting possible proteins to be involved, in particular in pancreatic cancer. We are concentrating on pancreatic cancer because on paraganglioma, obviously the knowledge is very scarce, whereas in pancreatic cancer we have more literature to study. So at this moment we are engaged with this in silico study to clarify effectively the mechanism of the antiproliferative activity we have seen in our MTT experiments. So concluding, We identified different compounds with our scaffold containing the benzothiazole scaffold. Among a large group of sulfonamide compounds, we identified this compound 1E that show a good antagonism on PPAR alpha, but its cytotoxic potential was only detected at high micromolar concentration in pancreatic and colorectal cancer cell lines. When we moved to amide compounds, we obtained this compound 2B, that was a moderate antagonist in transactivation assay, but its cytotoxic effects were relevant in pancreatic, colorectal, and also in paraganglioma cancer models, with an additional inhibition of clonogenic activity when we tested it in our paraganglioma models. And about the last compounds I've shown you, we can conclude that the introduction of an alkylic chain as a normal butyl in alpha position is detrimental to activity, whereas the introduction of substituents on phenyl ring were led to the discovery of different derivatives endowed with a good antiproliferative effect, in particular compounds bearing electron withdrawing substituents in meta and para. In our library of compounds, we selected compound 4D, which a trifluoromethyl substitution on para, that was the most cytotoxic compound, and the derivative 4L, which an acetamide in meta position that was less toxic against non-cancer cells. So at the end, I would like to thank all my collaborators at the medicinal chemistry group of my university, the University of Chieti-Pescara in Italy, with my colleagues who helped me in the synthetic part and the target prediction studies. And I would thank also the pathology group of my university for the biological studies on cancer cells. And I would thank all of you for your kind attention.
Thank you for your presentation. Now floor is open for the questions.
Thank you for the very nice talk.
Thank you.
So the first question is that when you try to modify your ligand, it seems that you always sort of restrict yourself to aliphatic chains or the aromatic groups. Is there any reason that you take this strategy?
Yes, we in previous work in similar compounds we both analyzed alkylic chain or aromatic ring with the aim to see if this modification could affect the ability to bind PPAR alpha. And in these experiments, we saw that both substitution, alkylic chain or aromatic ring could ensure a good binding to the receptor. This is the main reason to try both the substitution.
Yeah, thank you. And the second question is that at the end, you show that you have a modified ligand that works with cells, but you are trying to search for the target protein. So this is the part that I'm a bit confused, because usually people do it other way around, that you first have a structure of the target receptor and then you try to find the drug for it, isn't it?
Yes, it is different, this approach, in the sense that at first our target was the PPAR alpha, and we were our aim was to detect novel compounds acting as antagonists on these receptors. But then we changed our idea because we saw in our benzothiazole compounds that they were cytotoxic, but they didn't block in a sufficient way this receptor. So it is evident that now for this novel series of compounds, we are in the search of a target. The PPAR, the action on PPAR alpha couldn't be the target that justifies the action, the anti-tumor, the antiproliferative activity.
Yeah, thank you.
Thank you.
I can see one more question from the Dr. Bruce.
Yes, really interesting talk. At the very beginning, you talked about the molecules helping to change from glycolysis to fatty acid metabolism. And I'm wondering if you think this kind of therapy in conjunction with a ketogenic diet might be even more helpful for patients that are suffering from some of these cancers.
Professor, it is a difficult question to answer in the sense that we have no in-depth information about the possibility to have a therapy with these compounds. We are really at the first of the preclinical development. Perhaps they could be better, but it is difficult to give an opinion at this point of the research.
Okay, thank you so much.